Thank you. Hello? Okay. First, I'd like to thank the organizers and the committee, and also all the speakers for the great talk so far. Today, I'm going to be talking about, I'm looking at the neuroblastoma and breast cancer data sets for the Cancer Data Integration Challenge. I'm going to skip this for now. My goal is to look for genes or certain pathways that, or genes in particular, that have functional differences between different groups, so different, clinically different groups of particular cancer, and then so functional differences and also genes that have some sort of clinical relevance. And there are four basic parts to this. First, one key thing is that we're going to look at reactome pathways. So I'm breaking up this big, gene expression data set to only look within certain pathways. Within a pathway, we're going to infer the association network. And for that, we can use any association measure, but I'm going to use partial correlations, which we'll look at. When we compare two groups, there's going to be a differential network analysis to see how genes are changing or how the associations between genes change in two groups. And then we're going to use that information to fit a survival model, and that'll give us an idea of if these changes are clinically relevant. And at the end, I'll give an overview. So again, the functional differences we're going to identify by differences in association between genes based off of the gene expression profile. And clinical relevance, we're going to look at survival model. So this is just to summarize the two data sets that we have. The first one is the neuroblastoma. We have RNA-seq data for tumor samples, 498 tumor samples. We have overall survival times, and we have other clinical covariates. I decided to use the ISMB stages of the disease as different subgroups to look at. So these would be the different subgroups that we'll see later. For the breast cancer data set, we have also RNA-seq information, so gene expression profiles for almost 2,000 patients, overall survival times, and for that data set, I decided to look at the Claudin subgroups. Okay, so the reactome pathways. I just looked at pathways that had between 10 and 100 genes. Some pathways have up to 1,000 genes or even more. I just looked at these, which gave me about 1,500 pathways. And we're going to look at, I look at all those pathways, but I take one at a time. So for example, here's one pathway that I might look at. And within that pathway, there are 18 genes. And so this is the information that I'm getting from the reactome database. I'm just looking at the genes within a certain pathway. And from these genes, I'll subset the gene expression profile to learn the associations between those. So the association measure, you can use anything. This framework is fairly general. I'm using partial correlations. So partial correlations, if you model the gene expression with a Gaussian graphical model, edges in that model relate to conditional dependencies between genes. And you can calculate partial correlations using the inverse covariance matrix of the Gaussian graphical model. And actually, this is kind of a hard problem in and of itself. I'm using this package that uses a shrinkage estimator for the covariance matrix, and that allows me to invert it and estimate these. But I mainly picked this because it was a convenient package and it was fairly quick. But there are tons of different ways you can go about even computing or estimating the partial correlations. So that's a whole other talk. But again, this framework is general, so you can use any association that you'd like. So we'll look at an example. First, we're going to see neuroblastoma. I'm splitting up into the different INSS stages, so stage 1, stage 2, stage 3, stage 4, and stage 4S. And I have those 18 genes from that one reactome pathway. And this is it's kind of blurry, but these would be the associations that were inferred from the partial correlations. So if you see an edge, it means that the partial correlation between those two genes was non-zero. And we can do it for stage 2 patients and so on. Now, so that's association. And again, you can use any measure you want, and you'll get some sort of association matrix like this, or a network like this. So next, we want to say, okay, we have association within a group, but are there any differences in this association network between two different clinical subgroups? So for instance, we might look at stage 1 versus stage 2 patients and see if there are any differences in the connections. And if you look at this gene here, you can see that in stage 2 there are these connections, whereas those are missing here. So that might be something where this gene is having some different functional behavior in one group and not in the other one. So maybe it's for whatever reason. And that might give us some information. So to actually do this, we just look at average differences in the association scores. So I'm going to call this a DC score, so differential connectivity score. And you can look at any set of connections in the graph. So I can test to see if the entire pathway is differentially connected, in which case I would sum over all the edges in the network. Or I could test if a single gene is differentially connected, in which case I would sum over just the connections for that particular gene. Or I can test a single connection, in which case it would just be the difference of that association measure between the two groups. And then the differential network will be all of those connections that have a significant difference. So here I'm looking at p-values. And p-values come from a permutation testing procedure. So again, you can use any association measure. You don't need to know any asymptotic properties of it or anything like that. If you did, you could use some other way of doing hypothesis testing, but we're just doing permutation testing. So to determine if a set of connections are differentially connected, we'll do a permutation test. If the connections were the same in both groups, then that means that you can basically interchange samples between groups and recompute the association matrix, and you shouldn't see any changes. So that's how the permutation is done. You just randomize the group labels, re-estimate each association network, and then recompute the differential connectivity score. And then the p-value will just be the number of times that your observed differential connectivity score was greater than the permuted scores. So that's how you would do it for two groups. You look at all of the individual connections that are significantly differentially connected, and if it is, then you have a connection in that network. And then if you have more than two groups, which we're looking at 5 groups, right? Stage 1, 2, 3, 4, and 4S. You just take the union of each pair of networks. So you'll see it. Well, when we look at breast cancer, you'll see something that will probably make that clear. I don't, talks are kind of bad because I can't really interact, and I don't know if anybody's lost at this point or not. But I don't know. So anyway, here are the 5 different groups. And very, very faintly here, you can see sort of the light brown edge, where that would be a connection that had appeared in some other group, but not that group. And this would be the union of all those missing edges, or edges that one group might have and not the other one. So this is the differential connectivity network. So these are all associations that occur in one group, but not another group. Okay, and then the key thing from here is we're interested in the genes that have edges in this network, so that would be V star, the vertices, and the individual connections. And now I'm going to denote that as E star. That'll be used right now. So the next step is clinical relevance. So the way we're going to assess clinical relevance is, does that differential network contain information that will help us predict something about survival times? So what we use is just a simple accelerated failure time model. This is a linear model with respect to log of survival time. Here we have coefficients for each of the genes in the network, the differential connectivity network. And then we have interaction terms for the edges that occur in there. And this is a parametric model, so we can have maybe a log normal distribution or Weibull or some other parametric model. So now after you fit that, there are some questions. For example, how good is the fit? You can look at likelihood or mean squared error, or since we have censoring, you'd want to do a modified mean squared error. You can look at C index, which is our predicted times correlated with the actual times. This is a concordance index. And the other question is, are these interaction terms significant? If they're not, then we didn't really learn anything by including those. And we use a likelihood ratio test right now, but you can also look at AIC or BIC and compare two models, one with and without the interaction terms, and see if it improves the model or not. For neuroblastoma, we just looked at, once we fit that model, we no longer look at group labels. We just look at the entire data set, and for predicted times, we threshold the time, and we have two groups here with two different survival times. And you can see that there's a significant difference between these two groups. So this suggests that the survival model is telling us something about the data. And again, this is only based off of the 18 genes that we were looking at in that pathway. So how am I on time?
You're good. Yep. You still have like 20 minutes.
Okay. So a quick overview. What we do for each pathway is we estimate individual networks. We estimate the differential network. We fit the accelerated failure time model. And then we can look at log rank tests. So the way I ended up sorting the pathways is by the p-value for the log rank test and how well it did separating these two groups in the training set. But there are probably better ways to actually sort the pathways, and this is something that we're currently working on. It might be good to sort them based off of the overall fit of the model, or you might want to sort based off of how differentially connected the networks are. And at this point, I want to say another issue was that this model that we are using doesn't work so well right now. The fit typically isn't that good, but it's good enough to get these kinds of results. Okay, so looking at the same pathway, everything was fit. Just looking at neuroblastoma, and we saw that it told us something, that these results are pretty good. So we decided to look at the same pathway for breast cancer. This is the differential connectivity network for breast cancer, and we're looking at the different Claudin subtypes. So here are a few. Now, when you compare LUM-A versus basal, these would be the connections that are in one network and not the other one. If you compare these networks, these pathways are differently, or these associations are different, and then there are no differences between those groups. And essentially, this overall differential connectivity network is the union of all these individual ones, and there are a few more that I didn't show here. So maybe that helps kind of get a picture. So we're looking at pairs of groups of patients. These are the different connections, and then we just take the union. Again, we find some pretty good separation in the data. But if we also look within subgroups, this one here was significant within LUM-A. We were able to find subgroups within that subgroup, but none of these other ones were significant. And then another thing we might want to look at is how do those differential connections, is there any similarity between neuroblastoma and breast cancer? So these are the two differential DC networks overlapped, and then the bold lines are the ones that they have in common. So basically, these genes are differentially connected in both neuroblastoma and breast cancer. And this TP53 was one that came out actually in a paper this year, in some other pathway, not this one, that they're looking at for targeting for drug treatment. And okay, that's all. I think maybe I'll take questions and maybe clarify.
Thank you very much, Tyler. So we have time for questions. Please come to the microphone.
Okay. Comment and question. First, I like this approach very much. We were also looking for interactions between genes for predicting outcomes for cancer patients, but using less smart approach. I mean, we just did brute force interactions of all possible possible interactions between all possible proteins, which doesn't make sense. So your approach is actually much smarter. Although ours is simpler, so we can make it really fast. But then, unfortunately, the models that use these interactions were no better than models that didn't use interactions. So do you have any estimate for performance?
Right, so that's what we look at here. So we can look at...
I understand that these metrics, but the results, can you compare the results of this approach with something simpler that doesn't just you select genes and see differentially expressed genes and compare of the result? We just did, well, as I told, simpler but say brute force approach, and we didn't see any differences whatsoever between approaches based on just single genes and then interactions between genes.
Okay, so you're saying that if you exclude these interaction terms, you didn't find any differences in your models?
Well, I have slightly different model, but basically I'm also looking in the interaction.
Well, in our case, a lot of the times they weren't important. So you can drop these and you get same results. But in this case, this happened to be a pathway where if you include these interactions, the model was better.
Yeah, but you could add data from different pathway and then get this information back to the model. This is what we combined all, say, top genes that were present in the system, and then it were basically the same.
Yeah.
I really hope that I will see some interactions. So I just wonder if you can see with this smarter way, when you combine the data from entire set of genes, not on...
Yeah, if you combine pathways, I imagine these would become unnecessary if you combine over pathways. But part of the reason why we're even looking at this is to say that if we just look at this, these connections do have some information. So it also supports the fact that pathway is differentially connected. So it's evidence towards that. So, I mean, you can use other data outside that pathway and maybe get the same information, but this is also part of the goal is to support the fact that that pathway is differentially connected.
Okay, I'd like to talk with you later on.
Thank you. Dimitra, can you come to the microphone?
Yes. First of all, very nice talk. I admire very much the regulatory exploratory statistics. It's I think for now it's it's better theoretical background for developing any inferences for the moment. Let me say in future maybe machine learning will do help. But anyway, so first of all, I'd like to ask you about this permutation test. Do you use any resampling?
Right, it's resampling with replacement, or with
Replacements,
Yeah, so
okay.
you're just swapping the
The
labels.
swaps, yeah,
Mhm.
and this is the reason you use the permutation test. So after that, I have the similar question to Vito Trudnitsky about the interactions. So it's a very interesting topic, the interaction.
Mhm.
What is the statistical test you estimate the significance of the interactions here?
So what we do is we fit a model with and without the interactions. And we
Yes.
can take the likelihood, since it's a parametric model, we can look at the likelihood.
The
And we look at the
likelihood
likelihood ratio of
You
those
mean
two
the
models.
likelihood function?
Yes.
Likelihood
ratio.
Okay. Okay, fine.
And then,
So-
but you can also look at the AIC
No,
of
no,
each model
no, no,
and
no.
then compare, see if it's lower, if you include.
Yes, but as a test, the likelihood ratio is stronger than a Kaiken, stronger than Bayesian. Yeah, everybody knows that. So likelihood is likelihood. Okay. So, and the next thing is as well about these interactions. So have you tried another, let me say, way of testing, maybe a non-parametric or something like that?
Mhm.
Maybe because the interactions are very interesting. Definitely, we should, let me say, a bit restructure your data or reorder your data, but this is your, how to say, task to do. But I would like to listen to your idea if you apply some other, how to say, test of significance, so let me say, non-parametric approach. It will be very interesting to compare. So,
Right.
yes, this is
Yeah, that would be
that's
something good to look at.
yes, okay. So, and finally, let's go to the graph, the subgraphs at the end.
At the end.
Okay, so what is the significance in information between the real graph and the subgraph? Let
Well,
me say.
So all we look at is the combined graph. Like this is what we use when we fit that survival model.
Yeah.
You could just consider these separately.
Yeah.
Right now, I'm just showing you how this was constructed by looking at pairs, each pair of
Yeah.
subgroups.
Yeah.
And
Each pair is subgroup. This means the edge between two vertices.
each clinical subgroup, so like Luma
Yes.
A versus basal,
Yeah. Okay.
we look
Yeah.
at, these are the differential connections for those two clinical
so you
groups.
made only the differential connections. Okay. So, okay.
So you can say, okay, and these compared to, if you look at these two groups of patients,
Yeah.
possibly this gene has the highest degree, so maybe
Yeah.
this one is playing some role, whereas in these, it's not.
Good.
And overall, you can look at the degree in the overall
overall,
graph.
yeah, okay, yeah.
If you want to just say overall in breast cancer, this is there's some functional changes if you look at different groups. And these are just showing you where those differential connections come.
Okay. And my last question. So the most important thing in, how to say, bioinformatics or biostatistics in biology, this is validation.
Right.
What is your, how to say, approach or your idea to?
Oh, well, so right now, all we are doing is partitioning the data that we have into training and testing, but obviously we'd like to have an outside, like another separate neuroblastoma or separate breast cancer data set that
Yes.
we can run this on and see if we get similar connections.
To make a test of significance, definitely.
Right,
somehow,
so-
to compare them, yes.
We can do that for the survival model. We can
Yeah.
say we can easily see if that's still working on the new data set. But as far as the differential network goes, that's harder to validate,
Yeah.
I would say.
Yeah.
So that's something that we are still thinking about, how to validate
Yeah.
these networks, the networks themselves.
My hint to you is, I do not know, but I'm trying to do that, yes. If you develop the survival function as a probability model function, so, and after that to look for the, how to say, to validate it, how it works. Maybe it's okay, it's another view, okay. Okay, thank you very much,
Thank
thank
you.
you.
Thank you.
I'll ask you
Okay.
a question. I was wondering that when you rank the pathways based on, let's say, enrichment score or whatever, but when you rank the pathways depending on differential network score, are they similar or they are
So
different?
if you use different measures, they're almost never similar. You get different rankings. So one thing we might look at is some sort of rank aggregation to
Yeah.
combine them.
Okay.
But that's something that I'm also thinking about is
Okay.
what's the best measure to rank them by.
Okay.
And the other thing is that we are ranking on the measures based on the training data set. So, that's
Okay.
an important point.
One more question.
Yes.
For the modeling. When you have the interaction terms, are they just based on association between gene X, gene I, gene J, or they are sort of connected to the differential part of it?
So these are over the edges that appear in the differential connection network.
So it has to pass the significance of differential criterion.
That's right.
And that is on behalf of individual genes connectivity, how it's changing.
Individual connections, so not even
Individual.
the genes. So we look at each connection and do a permutation test. So we get a p-value for every single connection.
Right. And the connections are between genes, though.
Right.
Yeah.
So it has to appear here in order to
Right.
have an interaction term.
So it will depend on cutoff and all these things. So it could be a tuning parameter of the model.
Right.
Okay. Got you.
Yeah, I also have a question. So you selected a pathway based on the neuroblastoma data.
That's right.
And then you also compared how, for this specific pathway, breast cancer behaves. And for breast cancer, it was not as meaningful, I guess, for stratifying patients as it was for neuroblastoma. So have you also looked at some pathways that were basically ranked the highest directly in breast cancer?
No, we haven't gone that way yet. We started working on neuroblastoma, and we had results for that, and then we compared those to breast cancer. So next, we will look at breast cancer first and see if those rankings show us something good in neuroblastoma.
Yeah, I mean, it totally made sense because this way you could actually compare breast cancer to neuroblastoma, right? Looking at the same pathway. Cool. And one more question. You used reactome pathways. So have you also tried how this ends results if you use some other pathway database?
No, so far we've only looked at reactome. Initially, I was considering KEGG, but it was harder to actually get those data sets. So reactome was just the easiest to integrate into the software right now, and what probably we'll look at other pathway databases as well.
Okay, great. Thank you very much. Any other questions? Okay, there's one more.
One more question. When you inferred these connections between genes, have you done this multiple testing correction, or just took each test individually?
Well, right now I'm just looking at them individually, but I do have one constraint when I'm actually doing this. I make sure that the pathway is differentially connected, the gene is differentially connected, and the connection is differentially connected. So it has to pass many tests. So one thing that we'll probably do is run some simulations to see what the true discovery rate or what the actual type 1 error rate is. But right now, it seems that's something we have to think about. Yeah.
Okay, thank you.
Okay. Thank you very much.
No, it's fine. I will just check the timing from time to time.
Okay, so our next speaker will be also talking about data integration, cancer data integration challenge. And his name is Maarten Laumassou from the Ghent University in Belgium.